So, we are ready. Thank you very much, Eva, and hello, everyone. I hope you all are enjoying the symposium and the amazing talks. They are incredible. So now we are going to start with our first session of the work selected as oral communications. The first speaker is Ana Diaz-Ucera. She's a PhD student in health science at the University of La Laguna in Tenerife, and she's focused on the Canary Island population, next generation sequencing, and bioinformatics. Today, she's going to present her work.
I am Ana Diaz-Ucera, a PhD student in health science doctorate at the University of La Laguna. First, I want to thank the organization for considering my abstract for an oral presentation. Today, I'm going to talk about a little section of my current doctoral thesis, which I would want to entitle Whole Exome Sequencing of the Canary Island Population to Unravel Natural Genetic Variation. I would like to start with a short introduction to explain the importance of the Canary Island population in the context of European diversity landscape. The Canary Islands are a Spanish archipelago of 7 main islands located in the Atlantic Ocean, 100 kilometers off the northwest African coast. Recent studies have pinpointed that the Canarian genomes can be modeled as composed of the ancestry proportion, which you could see in the current slide. From 75 to 83% by European, from 17 to 23% by North African, and less than 2% by Sub-Saharan. However, one of our most recent published studies has shown that ancestry can be as high as up to 29.9% for North African and 9.2% for Sub-Saharan ancestry. Interestingly, African genomic ancestry has important biomedical implications for European population, and it's associated with risk for cardiovascular, renal, and respiratory diseases, as well as with diabetes, among others. Curiously, the prevalence of some of these diseases and/or their complications is higher in the Canary Islands than in other regions of Spain. This is the case of diabetes, obesity, and hypertension in all age groups, and of asthma and allergic diseases in children. Because among the European population, the Canary Islanders exhibit the largest proportion of African ancestry known to date, a detailed knowledge of the natural genetic variation in this population will allow to establish a reference catalog in which to sustain medical genetic studies, besides allowing to investigate its origin, recent history, and isolation. A total of 629 donors were recruited for the Princeton study. They self-reported absence of cardiovascular, metabolic, immunologic, and cancer diseases, as well as at least 2 generations of ancestors born in the Canary Islands. Additionally, they didn't declare family relationships among them. DNA samples were obtained from peripheral blood using a commercial column-based solution and implementing commercial whole exome enrichment capture kits. The different quality control were assessed by means of an Allele 4200 tape station system and a Qubit 3.0 fluorometer. Pools of insect sample at 2 nanomolar loading concentration were sequenced on an Illumina HiSeq 4000 sequencing system with 75 BP per end reads. BBBA-GATK version 3.8 best practices were followed for germline variant calling using the human genome reference in this slide. ANOVAR, EnsembleBep, and Interval tool were used for functional annotation, and discriminant analysis of principal component was implemented as population analysis. A total of 340,930 variants were identified by whole exome sequencing in the target regions, of which 70.1% were exonic. A quarter of them were absent from the 1000 Genomes Project, NOMAD, and TopMed. In this last type of variant, we could observe an allele frequency distribution as shown in the current slide, remarking that 97.4% of those present an allele frequency below 0.5%. When pathogenicity catalogued by Interpret tool was considered, 3,276 of them were classified as pathogenic or lightly pathogenic. From these non-benign variants, nearly 90% of those were loss-of-function variants or had high-impact prediction, being frame shifts the most common variant. By means of enriched web tool and a Phytep visualizer, an enrichment analysis was carried out on those genes which encompass high impact and high confidence loss-of-function variants at the same time. This analysis supported a clear enrichment in genes involved in oncological diseases such as kidney cancer, carcinoma, liver cancer, endometrial cancer, breast cancer, skin cancer, melanoma, and pancreatic cancer, as well as in intellectual disability and neurodegenerative diseases in top 10 more significant genes. When we move to population analysis, we could notice interesting patterns. El Hierro and Gran Canaria form two clearly separated and well-defined clusters for all allele frequency range. For the remaining 5 islands, two clusters could be observed, one encompassing La Palma and Tenerife, and another including the rest of the island, I mean Fuerteventura, La Gomera, and Lanzarote. However, on the lower end of the allele frequency spectrum, in rare variants, Fuerteventura clustered closer with Tenerife and La Palma, while La Gomera and Lanzarote remained more distant from this cluster. In this respect, discriminant analysis of principal components reflects a pattern that is reminiscent of historical episodes. Fuerteventura, Lanzarote, and La Gomera are the islands with the largest proportion of North African ancestry, whereas Tenerife and La Palma were known as manorial islands in the historic time. Therefore, the conclusion of the current talk are: first, approximately 25% of the detected variants were novel, and 97.4% of those presented an allele frequency below 0.5%. Second, discriminant analysis of principal components revealed genetic differentiation among islands, being more marked when rare variants were considered. And finally, based on the present results, the need for a catalogue of genetic variation in the current Canary Island population is more evident than ever. I would like to end by thanking again the organization for this oral presentation, as well as my thesis supervisor for the opportunity, and all the entities that finance this project. Thank you so much for your attention.
So that's it. And now we move on to the round of questions for Anna. Anyone has any question?
Hello, I'm here.
Yeah, I see one. Let me check.
Okay.
Here, Javier Lanillos asks, congratulations for your work. Do you plan to submit detected variants in a repository?
Okay. Yes, it's it's one of most is one of the things that we know we have to do, because actual current science is based on the share of this kind of information, but we are working in increase the number of donors for this cohort. So when we have got the final cohort, probably we submit this these files to our repository, maybe a GitHub or own website. Yeah.
Nice. We are waiting for another question. Here we are, another one. Congrats do you think it will be any difference with whole genome sequence data?
Yes, really, it's the different technologies supply a different variation, different analysis. We are going to base our I'm developing my thesis in the exome analysis, but there are another mates that are working in the exome in the genome context, because we have we know that the most multidisciplinary catalog is the basis for a medicine personalized project. So in our final catalog, indeed, we are make a kind of hybrid with array data, whole exome sequencing, and whole genome sequencing. In this case, I talk about my thesis that encompass the exome analysis. Sorry if I a little nervous. I spend a lot of time without talking English, so I'm a little
Don't
nervous.
worry. I think everyone has the same problem. So here we are, another one from Miguel. Very clear presentation, thanks. If I understood correctly, you mentioned that African ancestry is correlated to some diseases. How much of these genetics bias different socioeconomic relatives?
If I understand the question, well, I don't know if the different socioeconomic realities is affecting. That I know is there are different studies that support this correlation between North African ancestry and the diseases that we can see more prevalent in Canary Island. The fact is that in Canary Island, apart from the different diseases or the different traits that are explained in the presentation, we have got a prevalence of the different diseases so much higher. We have got a prevalence of the diabetes that it's 2 times higher than in the Canary Island, both the mortality and the morbidity. So I can talk about the genetic context, but no for the socioeconomics reality.
Yeah, so I think that's a very good answer. Well, we have time for one more. I don't know if someone wants to ask something else to Anna. If not, I can just make the last one. Just for curious, how do you explain in the presence of pathogenic or likely pathogenic variants in a healthy cohort?
Sorry, can you repeat?
How do you explain the presence of pathogenic or likely pathogenic variants in a healthy cohort? Just for curious.
Well, I know that it's a possible question because I was the first person that surprised by this situation, but we have to think that have a pathogenic variant doesn't mean you suffer or express the disease. You have got into account a lot of different parameters. The zygosity, if you have the variant in homozygosity or heterozygosity, if this if the pattern of heritability is recessive or dominant, maybe even the environmental exposition. So you have got you may got a pathogenic variant, but you may not suffer the trait. Probably if we select we have seen this in this cohort, we selected healthy donors and we see different pathogenic variants. So being health doesn't mean you don't have pathogenic variants.
So perfect, Anna. Thank you very much. We are going to the next talk now. Now we continue with Patricia Sanchez Jimenez. Patricia is also another PhD student whose research field is drug-resistant epilepsy in adults undergoing neurosurgical resection of the epileptogenic area, through a transcriptomic approach. She looks for differentially expressed genes involved in the pathological mechanisms of drug-resistant epilepsy. So today she's going to present her work. A minute, sorry. Here.
Patricia Sanchez. I am talking about transcriptomic exploration of epileptogenic zone of drug-resistant temporal lobe epilepsy patients. First of all, I would like to introduce you what is epilepsy. International League Against Epilepsy, an expert committee, set the basis of the disease. They defined epilepsy as a disorder of the brain characterized by an enduring predisposition to generate epileptic seizures and by the neurobiological, cognitive, psychological, and social consequences of this condition. And the definition of the epilepsy requires the occurrence of at least one epileptic seizure. An epileptic seizure is a transient occurrence of signs and/or symptoms due to abnormal excessive or synchronous neuronal activity in the brain. About the impact of epilepsy in the world, it is relevant to know that it affects 50 million people worldwide. 80% of the patients live in the low-middle income countries, and of them 75% don't receive treatment. For that, epilepsy increases 3 to 6 times the rate of premature death. They also suffer stigma and discrimination. And if we focus on Spain, there are over 400,000 patients with epilepsy. 30% of the patients don't respond to anti-epileptic drug treatments, and they are considered as drug-resistant epilepsy patients. Allele defines drug-resistant epilepsy as a failure of adequate trial of two tolerated and appropriately chosen and used anti-epileptic drug schedules to achieve sustained seizure freedom. DRE is an acquired disease and it is independent of the drug treatments once it appears. It may show temporary remission period, but epileptic seizures frequently reappear. Moreover, the prevalence of DRE is higher in temporal lobe epilepsy patients. Furthermore, the molecular mechanisms of drug resistance are still unknown, although there are many theories that try to explain how patients change from pharmacosensitive to pharmacoresistant. However, there are other therapy options for these patients, such as surgery or diet. There are different kinds of surgical intervention, being the first option the resective surgery of the epileptogenic zone. Indicate that it would be possible, there are 50 to 80% of chance of achieving seizure freedom. Where this is not possible, other types of intervention can improve the probability of eliminating or reducing epileptic seizure. However, when surgery isn't a therapeutic option, diet may be a good alternative. For example, ketogenic diet is suitable in children and patients with some epilepsy syndromes. Thus, considering all the above, our main objective is to understand the molecular mechanisms involved in drug-resistant epilepsy patients. For that purpose, we selected a cohort of 47 patients who were diagnosed with drug-resistant temporal epilepsy and subjected to the resective surgery of the epileptogenic zone. All of them from Hospital Universitario de la Princesa in Madrid. And we also selected a cohort of 37 postmortem brain donors to the Spanish National Biobank Network, especially from these 4 biobanks. All the participants had never suffered any biological or neurodegenerative disease, and they were paired by age and sex to our patient cohort. We performed an RNA-seq study with FFPE hippocampus of patients and controls. We sliced logs and extracted RNAs from them. Then we made quality controls and quantifications. We prepared cDNA libraries. We performed another quality control and quantification. After that, all samples were pulling together to get the sequencing pool. It was sequenced to an average depth of 100 million total per end on Illumina NovaSeq platform. Finally, we performed bioinformatics analysis, so raw reads were aligned to reference human genome, and read counts were analyzed by differential expression analysis to get the differential expression genes. Before explaining to you our main results, I want to point some characteristic data of the participant in this study. How I say above, in this project participate 47 patients and 37 controls. This cohort has similar mean age and number of men. Before surgery, our patients suffered a mean of 10 seizures per month, and 8 patients had more than 12 seizures per month, until 30 or 90. Almost half of the patients were suggested to right hemisphere surgery. After 2 years of the neurosurgery, 85% of the patients get Angel 1 state, and 68 of them were seizure freedom. Only 3 patients achieved Angel 4 state. These numbers evidence the success of the temporal lobe receptive surgery. On the other hand, we registered the adverse effect of the neurosurgery, such as depression, migraine, apathy, nausea, among others. The main output of this study was the differential expression genes, DGs, which are generated by comparing the expression patterns of the patients to the controls. We found 8,733 genes, and almost 4,000 were upregulated and more than 4,700 downregulated. Here you can see a short fragment of a large table that bioinformatics analysis generates, where we can see the gene symbol, ensemble ID, the log 2-fold chains, and the p-adjusted value after fold discovery rate tests were applied. This indicates the differential expression between patients and control, and that shows us how significant this difference is. We performed 2 enrichments to know in which cellular processes and pathways are involved. In the gene ontology enrichment, we found some interesting geoterms, such as the regulation of synaptic plasticity, modulation of chemical synaptic transmission, and dendrite development. On the other hand, in the pathway enrichments, we found that 69 of our DGs were involved in axon guidance pathway, and 41 in Fc gamma R-mediated phagocytosis. We also performed the validation of our RNA results. For that, we selected 7 digits and we made a real-time qPCR. For that, we selected these 4 genes that were regulated and these 3 genes that were unregulated. Thus, we confirmed the result that we obtained applying the transcriptomic approach. Finally, I sum up this study in the following points. We found 8,733 differentially expressed genes in drug-resistant temporal lobe epilepsy patients compared to controls, of which 3,959 genes were upregulated and 4,774 genes were downregulated. We found that digits are enriched in geoterms related to the brain, such as synaptic plasticity, regulation, and dendrite development. We also found that digits are enriched in pathway, for instance, axon guidance. We validated 7 digits of our RNA-seq result by RT-qPCR. For that, we selected these four genes upregulating at these 3 genes unregulated. Finally, I want to acknowledge all the people who take part in this project and institutions, and of course, thank you.
So thank you very much, Patricia. It was a great talk. So now we have the question for Patricia. Anyone wants to ask something? We can wait a little bit. I have one, just for curious, where we are waiting for the attendance. There are another therapeutic options for epilepsies, just for curious also.
For general epilepsy, there are so many drug treatments, but for drug-resistant epilepsy, there are a few. In the presentation, I said that surgery, in the case that it indicated, or diet, but I don't know if there are other treatment, but there are few for drug-resistant epilepsy patients.
Okay, thank you. And also, did you perform? I saw that you perform validation and also an enrichment analysis, but did you perform an in silico functional analysis for the most significant genes, just to see what is its relation with the disease and how it can work and something like this?
No, we don't perform it because we want to explore the RNA-seq, the results, and we won't know if, firstly, if the genes are related with the brain processes and what are there. But it's a great branch to explore.
Okay, thank you. Now we have one from Javier Lanillos. Amazing talk. Thanks, Patricia. Does your data allow you to check for transcript isoforms analysis? Any of your genes found could have different isoforms?
First, thanks for the questions. No, we don't check the isoforms analysis because we perform an RNA-seq of FFPI samples, and if you don't work with them, FFP broke broke all the transcript, and it's so difficult to reconstruct and to find isoforms.
Yeah, that's true. So I don't know if anyone wants to ask another question for Patricia. So, well, we can continue. Thank you very much, Patricia. A very nice talk and presentation.
Thank you.
Now we have Javier Lanillos. Javier is a bioinformatician in genetic medicine focused on pharmacogenomics and cancer research. And today he's present to us his work. Wait a minute. Here.
Hi everyone, my name is Javi and I'll talk about the use of diagnostic exome sequencing data for preemptive pharmacogenomics. Pharmacogenetics studies the variability in drug response based on the individual's genetic background. This field aims to avoid adverse reactions to drugs and increase therapeutic efficacy. The Clinical Pharmacogenomics Implementation Consortium, CPIC, establishes pharmacogenetic clinical guidelines. They define the important alleles in each gene, what pharmacogenetic phenotype they confer, and then the therapeutic recommendation on the use of the drug according to such phenotypes. For example, it's important to test few alleles on the DPYD gene, whose enzyme metabolizes fluoropyrimidines, which is an anti-cancer agent, and determine whether an individual is a normal, intermediate, or low metabolizer. After that, they can make some recommendations to adjust the dose. Pharmacogenomics can be very useful if we see that 7% of hospitalizations are due to adverse reactions to drugs. and that is estimated that more than 90% of the population carries at least one pharmacogenetic allele. This is a made-up scenario with different individuals and diverse medical needs. Pharmacogenomics can help with improving with issues such as therapeutic adherence, or deal with population diversity, population genetics diversity, reduce the number of hospitalizations due to adverse reactions to drugs, and also it can act on widely prescribed drugs or help to deal with polymedication scenarios, very common, for example, in the elderly people. So on the one hand, pharmacogenomics can be applied in a reactive manner, testing specific variants in situ and treat a patient with safety. But there is also the use of preemptive pharmacogenomics. For patients who underwent a genetic test using NGS technologies, why not using this data with a preemptive purpose that will be very useful, for instance, in the future? So this idea is getting more and more attention, as you can see here on the right. However, these studies are missing some genes and variants from CPIC guidelines. And therefore, all preemptive pharmacogenetic information that can be repurposed from exome data is not maximized. Our objective then was to perform a systematic analysis of the actionable pharmacogenetic variability in a large cohort using diagnostic exome sequencing data, and provide with the landscape that will guide the preemptive use of pharmacogenetics. So we used an exome sequencing dataset of 5,001 samples. 4,002 were whole exome, and 999 were sequenced with clinical exome panel. So variant calling data was crossed with CPIC guidelines information using Python scripting. And then this is a cohort of patients that comes from individuals who needed at some point clinical diagnosis for germline diseases, either in Spain or other countries in Latin America in this case. The workflow here is that you can see here describes our gene selection, what genes can be actionable using exome data. So we first filter out those alleles who actually don't confer an actionable function, such as normal, uncertain, or limited evidence alleles. After that, we filter out those alleles which are not really covered by exome sequencing technology. And at this point, we reasoned that if one important allele had been filtered out in the previous steps, the pharmacogenetic report for such gene would be incomplete and therefore not suitable for the clinics. After this step, we found out that 11 genes can be suitable for analysis using exome sequencing data. Of note, 99.3% of the variants that we tested were covered at 20x coverage. In 5,001 individuals, we found that an average of 2.2 actionable alleles per individual, so that 94.2% of the cohort carried at least one actionable allele. If we look a summary at gene level, UGT1A1 was the most prevalent gene, followed by genes such as CYP2B6 and 2C9 that show also a very rich heterogeneity in the number of different alleles found for each gene. Also, on the other hand, CYP4F2 and NUT15, they are represented just by one allele, and other genes are very low prevalent and population specific, such as G6PD and RIR1. Translating those alleles into the actual phenotypes, as expected, 90% of the population had at least one actionable phenotype in one gene. This, in result, accounts, or it's very important to point out that using exome data, these 11 genes can fully report 9 clinical guidelines at the end. So I also want to point out the importance of having pharmacogenetics data from different and underrepresented populations. Here, there is an example that the Spanish Medicine Agency recommends to test 4 alleles in cancer patients who require fluoropyrimidines. So these are the 4 most frequent pharmacogenetic alleles. However, we found, for example, a 5th allele that whose frequency is comparable to them. In conclusion, we made a systematic analysis of CPIC guidelines and maximized the number of actionable genes and alleles that can be informed and used for preemptive pharmacogenomics. Of note, whole exome and clinical exome panels fully inform, they perform similarly and fully inform 9 out of 25 CPIC guidelines. In our cohort, 93% of the individuals carried at least one phenotype. And it's very important to repurpose exome data to help characterize the pharmacogenetic landscape in each country. This way can help experts to suggest different strategies and choose what alleles are more important to test in different countries and help to develop, for instance, personalized medicine initiatives. In the bioinformatics part, I think the most important part now is bioinformatics automation. So making pipelines and tools that can help to extract all this information in a robust manner from large cohorts so that we can help boost pharmacogenomics. So there are here some very important tools that are being developed. And also it's important to make more effort to characterize indel variants so that we avoid false negatives, for example, from our data sets. Finally, I think whole genome and long-read sequencings are possible solutions to solve some of the genes and alleles that have copy number variants, or they are the HLA genes, for example, so that more clinical guidelines will be informed based on the repurposing of NGS data. So finally, I first want to say thank you to my group, the Hereditary Endocrine Cancer Group at the CNIO, especially my thesis supervisor, Cristina Rodriguez, and our outstanding collaborators at NIM Genetics, Sara, Paola, and Marta. They helped us so much. It's been very nice to work with them. And thank you all for listening.
So really nice and really interesting talk, Javier. I don't know if someone wants to ask something to Javier. This is your moment. We can wait a little bit, but I can ask you one question. We are waiting. So as you mentioned, indel variants in your conclusion, why do they need better characterization?
Thank you very much for the question, Tamara. The presence, we have found that indel variants for pharmacogenomics are very important because in some genes they are highly prevalent in the population. So we will find, for instance, in the UGT1A1 gene that half of the population will have a specific variant. So, and it's an indel variant. So we need to recover that one if we can from exome data. But sometimes we have, for instance, that gene, that variant, it's in a homopolymeric region. So it's a deletion or an insertion of 4 or 5 letters in a repetitive mode, which sometimes the pipelines are confused and they make false negative errors, which we don't want. We want to avoid. That's one of the first issues that I haven't seen like a lot of effort on indel variants in these papers that I mentioned. And also, the other part of the indels is that sometimes variant callers, they call indels differently. So they are the same indel, but they are not annotated the same way. So it can confuse the subsequent analysis. So sometimes what they do is normalization. So you try to normalize all the indel variants so they all look the same. And that is something that is recommended, but still not, there's not a robust way to do it. And I have encountered some problems with that.
Okay, nice. Thank you for your answer. We have now one question from Sara. Do you have any ideas on how we can encourage physicians to adopt and apply CPAC guidelines in clinical practice? CPAC is a great resource, but my understanding is that its application in clinics is not as high as it could be.
Yeah, that's what we all want, that physicians really adopt the use of CP guidelines and pharmacogenetics. So from the bioinformatics point of view, what we can do is make everything so easy that they just have NGS data, they have the ethical framework, and then we take care of the technical part. And we just report a very simple table saying this gene, this person has this variant in this gene. And we recommend that in case you have to prescribe this specific drug, be careful, because there is a CPIC guideline saying that you have to reduce or avoid that drug at all, so that you don't want your patient, for instance, coming back with an adverse reaction to the drug. So I would say from the bioinformatics point of view, that all the technical stuff behind that makes life really easy for them. We don't want them to have to interpret a lot of new stuff or be careful. And then, of course, collaborating more with the clinics. That's, I think, my main point with the PhD. Yeah, I'm learning a lot in bioinformatics, but at the end, what I really want is to help in the clinics. So thank you for the question.
So thank you, Javier, and thank you, Sarah, for the question. Now we have another one from Miguel. Thank you for the talk. How difficult is to include interaction between variants into consideration for pharmacogenomics? Sorry.
By interactions between variants, for instance, I kind of understand that there could be a couple of variants in the same gene, for example. So a combination of them, that's an issue. And sometimes you need to figure out whether two variants, for instance, they are in the same allele, so they are in the same haplotype, and therefore you have a wild-type allele and another allele that it's carrying both variants. So it's not the same having them in different DNA strands, for example. And also, yeah, perhaps a couple of genes can have two different variants that really matter for one drug. So it's how difficult is it? Well, using CPIC guidelines, they describe very well combinations between different genes and variants. So I think it's very clear, the message from the experts that really write down the clinical guidelines. And if we get into the scenario with the variants interacting with each other, then I think most of the cases, they are well explained, and we can make a really easy explanation to the user. And on the other hand, there is this part that I didn't talk about, which is rare variants. Rare variants are very important as well. If you get a loss of function variant in a gene that is not described in CPIC guidelines, how do you deal with that? Because they are very important also in pharmacogenomics. So that's another field that is currently on the progress.
So perfect. Thank you very much, Javier. I don't know, I think there's not more questions. So yeah, we can continue with the last talk. Thank you very much again. And now we finish this session with Blanca Rodriguez Fernandez. Blanca is a PhD student at the Alzheimer Prevention Program Barcelona Beta Brain Research Center, and she's interested in neurogenetics and bioinformatics. Today, she's going to tell us about her work.
Hello, everyone. I'm Blanca Rodriguez, and I'm a PhD student at the Barcelona Beta Brain Research Center. Today, I will be presenting our last work entitled Genetically Predicted Telomere Length and its Relationship with Major Neurological Diseases and Life Expectancy, a Mendelian randomization study. Although neurodegenerative diseases have diverse clinical manifestations, they do share common features, mechanisms, and risk factors. One of the most relevant risk factors affecting these diseases is aging. As life expectancy rises worldwide, the prevalence of neurodegenerative diseases also increases, imposing a heavy socioeconomic burden on patients, families, and communities. So, given the increasing prevalence of aging-related diseases, in the last decade, the scientific community has made an effort to identify biomarkers of aging, which could predict better an individual's functional capabilities and chronological age does. In humans, telomere length is considered an objective biomarker of biological aging, since it shortens with age at similar rates across the tissues. Indeed, numerous studies have reported associations between observed shorter telomeres and higher overall risks for mortality, as well as with increased rates of age-related chronic diseases such as immune dysfunctions, cardiovascular diseases, or cognitive decline. However, telomere length and these outcomes are affected by common non-genetic risk factors related to lifestyle. Among other reasons, these common factors may lead to confounding and reverse causation from which observational studies are vulnerable. The Mendelian randomization approach was developed to overcome these limitations as a powerful tool to address whether these kind of associations are causal or not. Unlike observational studies, Mendelian randomization approach does not explore this association directly, but through genetic variants such as single nucleotide polymorphism, which are used to estimate telomere length in a way that is not affected by this confounding. Taking into account all this information, our main objective was to test whether telomere length may causally affect life expectancy and the risks of developing some common neurodegenerative diseases. To this aim, we used the summary statistics data of the most recent GWAS for telomere length, our exposure of interest, and our outcomes, comprising Alzheimer's disease, Parkinson's disease, frontotemporal dementia, amyotrophic lateral sclerosis, semantic dementia, progressive supranuclear palsy, and life expectancy. We saw observational analyses are prone to confounding and reverse causation. To avoid this, Mendelian randomization analysis uses genetic variants as instrumental variables to predict exposure of interest according to the carrier chief of the effects alleles associated with the exposure. For a genetic variant to be considered as a valid instrumental variable, it must satisfy the following assumptions. First, the genetic variant must be associated with exposure. Moreover, it must not be associated with the outcome due to confounding. And finally, it must only affect the outcome via exposure. So, if these three assumptions are satisfied for J genetic variant, the causal relationship between the exposure and the outcomes can be estimated by applying the two-stage least square method. In the first stage, we regress the exposure of interest on each genetic variant. And in the second stage, we regress the outcome of interest on each genetic variant. As a result, we obtain two sets of beta estimates, which represent beta X, the genetic association with exposure, and beta Y, the genetic association with the outcome. If a genetic variant is associated with the outcome only via exposure, this genetic association with the outcome is equal to the genetic association with the exposure multiplied by the causal effect of the exposure on the outcome. Thus, we can easily estimate this causal effect by applying this ratio. Moreover, the estimated ratios for each genetic variant can be combined into a unique estimate for all the genetics variants using the inverse variance weighted method. This schema represents the Mendelian randomization model that we apply in the present study. First, we obtain the beta X estimates and standard errors from the summary statistics of these GWAS. On the other hand, the genetic associations with the outcomes, or beta Ys, were extracted from the GWAS for neurodegenerative diseases and life expectancy. Finally, we apply the inverse variance weighted method to estimate the causal effects of genetically predicted longer telomere length on our outcomes of interest. As you can see, we have now included in this schema the alpha term. Alpha represents the potential pleiotropic effects. Pleiotropy occurs when a genetic variant affects the outcome through other unexplored pathways. However, the inverse variance weighted method assumes that there is no pleiotropy. So we apply the Egger method as sensitivity analysis to ensure that these intercept terms were close to zero for all the genetic variants. To summarize, you can see the Mendelian randomization method as a three-step procedure. Beyond the selection of the genetic variants that you will use as instrumental variables, the crucial step of the method. Then, you will only need the summary statistics data to estimate the causal effect of the exposure of interest on the outcomes. This table contains the results from the Mendelian randomization analysis. We found genetically predicted longer telomere length was causally associated with a reduced risk of Alzheimer's disease and increased life expectancy. However, there was evidence of pleiotropy for Alzheimer's disease results. Finally, we didn't find any other significant associations with the remaining neurodegenerative diseases. Previous Mendelian randomization studies have reported protective causal associations between longer telomere length and Alzheimer's disease. On the other hand, only 2 additional studies explored the potential association with amyotrophic lateral sclerosis and Parkinson's disease, with no significant results. Finally, no previous Mendelian randomization studies have explored the causal effect or the potential causal effect of telomere length on frontotemporal dementia, semantic dementia, progressive nuclear palsy, or life expectancy. Although aging is considered an important risk factor for most neurodegenerative diseases, we must consider that other biomarkers of aging, such as genetic instability or epigenetic alterations, might be leading these associations at the molecular level. So this is definitely a limitation of our study. Even so, our results support the protective role of longer telomere length on life expectancy and Alzheimer's disease, but no other neurodegenerative diseases. On the other hand, the presence of pleiotropy may reflect the complex interplay between telomere length homeostasis and Alzheimer's disease pathophysiology. So, further observational analyses are guaranteed to confirm these results. Finally, I would like to thank the Spanish regional student group for the opportunity and organization of this symposium, and obviously to the rest of the team at the Barcelona Beta and every participant of the Alpha study. Thank you very much for your attention.
So, hi again.
Hi.
Hi, Blanca.
Sorry for
Thank
this
you.
silence part. I don't know what happened, but
Don't
hope
worry.
you enjoy anyway.
I think it was very clearly, I think. And yeah, it was a very good explanation about the randomization studies, because I think it's very interesting and very common and popular now, I think, with the disease.
Yes,
For example, in sepsis, there are few studies from this, and it helps to identify new therapeutic option, or new variants that you can see with the other approach.
Yes, definitely.
So great talk. Well, now I don't know if someone wants to ask something. Yeah. Javier Lanillos said, "Thank you, Blanca. Very cool work. Will the length of telomere at other tissues be relevant for your work? And are you considering using UK Biobank data, which has 0.5 million people?"
Yeah. Yes, regarding the first question, it's true that this is a limitation of most US studies regarding telomere length, because they are mostly measured at leukocytes. So it's definitely a limitation, but it's true that an article published last year with DTA's data shows that the really telomere length and telomere length attrition, more important, correlates really well across tissues, including brain. So yes, it's good to consider this aspect anyway. And obviously, this work was done before the UK Biobank data regarding telomere length, the US, with almost 500,000 of people, was published with have access to the preprint. And yes, I don't know if we are planning to repeat the analysis using this data, but I will use this data definitely in other analysis of my thesis.
Nice. Now we can wait for another question. Come on, just make question for Blanca. I saw that the shorter telomere are associated with a protective effect for the Alzheimer and everything. Is that right? Maybe I missed something.
Sure. Yes, it was quite confusing and
Okay.
my fault. It was like we estimate, or we take as reference at the effect alleles associated with longer telomere length. So we found
Okay.
that longer telomere length has a protective effect on Alzheimer's disease risk in this case. But
Okay.
well, it's anyway.
Okay,
Yes.
now
Yes.
I understand. Thanks.
Thank you.
Here we have another question. Nice talk, Blanca. Have you considered exploring other outcomes variables such as gene expression, CPG, methylation, etc.?
Other outcomes, we are planning to add to this study outcomes related to the pathology of, for instance, in this case, we only found statistically significant results on Alzheimer's disease. So we are planning to use GWAS data on amyloid beta deposition and phospho-tau, just to check and also to study it in more depth this pleiotropy that we found, but we are not planning in currently to explore on others, CPG or
maybe polygenic risk score. You can also try to
yes.
see the relation between the
Yes. In my thesis
long-
that I work in alpha study, we have this polygenic risk score for telomere length, Alzheimer's disease, and other risk factors, and we are planning to use them. And also important to use them to categorize our individuals.
Okay, nice. Thank you.
Thank you.
I don't know if someone else wants to ask something to Blanca. It's the moment.
Come on.
So if not, we can conclude this session here. Thank you very much, Blanca.
Thank you.
So thank you. This has been the last talk with this session. And thank you very much to all the speakers, Blanca, Ana, Patricia, and Javier, for being here and for having presented your amazing works. And to all the attendees for listening and to ask the questions. So with all this, we conclude this session and we go to the lunch break. See you back in an hour.
See you. Thank you very much, everyone.
Thank you.